Parkinson's disease. Do you realize it's been 200 years since Parkinson described the disease named after him? We still really don't have an idea about what causes it. And it's been 100 years since Alzheimer's described that disease. And we also don't know what causes that, let alone have an effective treatment. We also don't really understand the mechanisms of autism, schizophrenia, depression.
Well, what was shocking to me was, A, they have never figured out what causes brain cancer. There's no known cause. I mean, and that hasn't changed in 25 years. And the treatment, the standard of care treatment, has been the same for the last 25 years. So there isn't a lot of options.
It's astounding that this is our health. This is our children's health. And despite hearing that cures for disease are just around the corner, if we actually look at the statistics, the situation's getting worse instead of better. The situation's getting worse instead of better.
The 50s was a time of growth. Suburbia flourished, the baby boom emerged, the economy expanded, and we witnessed a burgeoning medical field. New medicines, innovations, and the promise of cures and effective treatments.
Here, a magical drug, terramycin, in crude form.
The number of cases of polio recorded fell from 60,000 to 2,000 within a year of Salk's vaccine.
The X-rays are focused on the cancerous tissue. X-rays help destroy cancerous cells.
The excitement and promises of cures and treatments continued into the 60s, 70s, and 80s. As we moved into the 21st century, we continued to hear about breakthrough advancements. However, are these messages simply being repeated?
Cancer is going to strike 1 in 2 men and 1 in 3 women. So we are not exactly winning the war on this disease. One of the reasons is that our system for developing drugs for cancer is essentially broke.
We don't have effective treatments for most of the diseases like heart failure, diabetes, cancer, stroke, ALS, Alzheimer's, Parkinson's.
The truth is, treatments for major diseases haven't improved in 30 years.
Pathologies show that it was glioblastoma multiforme, which is a stage 4 brain cancer. So it's worrisome when you say, okay, you're going to go on chemo. You're like, well, what if I don't tolerate it well, or what if it, what's next? Well, no, there is no next. That's the one and only.
A few days after I had my baby, I was told that I had osteosarcoma, chondroblastic osteosarcoma, so bone cancer of the maxilla.
I first discovered I had Parkinson's was shortly after an injury to my shoulder. I noticed that I had a little twitch in my finger and my pinky on the left-hand side.
After a week of ignoring abdominal pain, that night at the emergency ward, they did an ultrasound, and I was given the results that very night. And I was told by the physician that it wasn't kidney stones or a kidney infection, in fact, that it was cancer. My right kidney was actually one massive tumor, and that the cancer had metastasized. It was out of the kidney and into the vena cava and had spread into my lymph nodes.
I had gone for my yearly mammogram. It's just something I'd been doing for the previous 10 years. And I got a call back. They said that I needed to go for a biopsy. I found out within the week that it was invasive ductal carcinoma.
I've had DBS surgery. It's called deep brain stimulation surgery. And it's where they take a pacemaker They put it under here, and they run a wire up all the way around the back of my ear, up here, and then they drilled a hole in my head.
Well, chemo actually nearly killed me more than once. I assumed that things were a little bit different since Terry Fox's time. I mean, that was 1980.
I made it through to the third treatment. However, I suffered one of the known side effects, and that was a near fatal attack on my liver.
I also qualified for a medical trial. So I went to the hospital every 2 weeks for an IV infusion of an immunotherapy drug. And I did that for 6 months as well. But unfortunately, in 6 months, during my routine MRI, it showed it came back.
It's more about taking care of yourself and finding the resources that are out there to help you. So yeah, I'm thankful for my surgeon, I'm thankful for my oncologist. They did what needed to be done, but post-treatment, it's definitely just up to us.
Opdivo can cause your immune system to attack normal organs and tissues in your body and affect how they work. This may happen anytime during or after treatment is ended and may become serious and lead to death.
Diarrhea, severe stomach pain or tenderness, nausea or vomiting, rapid heartbeat.
Nuplazid can increase the risk of death in elderly people with dementia-related psychosis. When taking Plavix alone or with some medicines, including asprin, the risk of bleeding
You have unusual chitta. Taking it with NSAID pain relievers, aspirin, or blood thinners may increase bleeding.
So when I look at the fact that thousands of people die from using a prescription drug as prescribed, we know we need much better regulation of the pharmaceutical industry, and we need much better information on those drug trials that even after passing Vanessa's law in the 41st Parliament, which was supposed to mean that we would have access to drug trials and we'd have mandatory reporting of adverse side effects, the Health Canada bureaucracy has been strangling the effect of what we did in the last Parliament. Big Pharma needs to be much more transparent. Canadians need to know that the drug they're receiving in a prescription is actually tested and safe, and we don't know that right now.
In making this documentary, I've learned that parts of our medical system are defective and outdated. To begin with, pharmaceutical and private companies often won't share information, which holds back progress. A vast majority, upwards of 50% of scientific discoveries published in medical journals are not reproducible. This is known as the reproducibility crisis. Furthermore, most animal data does not apply to human biology. Negative results are generally not published, wasting years of time and billions of dollars on retesting. Antiquated laws force scientists to follow unnecessary procedures, wasting time and money and hindering innovations. And alarmingly, 95% of drugs that pass animal testing fail in human clinical trials.
For over 50 years, the same biomedical system has existed. Yet, ALS, diabetes, Alzheimer's, most cancers, Parkinson's, and virtually all major diseases remain uncured with no effective treatments. And one of the major reasons is that government, charities, and universities continue to invest in an area that has a 95% failure rate. Although animals have helped in the past, human biological innovations are the future.
The rate of congruence between animal models and the human condition is at an all-time low. 95% of drugs that have been tested to be safe and effective in animals fail in human clinical trials. We're not 70-kilogram versions of mice, rats, rabbits, guinea pigs, cats, dogs, pigs, or monkeys. Human biology is very different from animal biology. Yes, we have learned from animals, but we have not been able to take that information and apply it successfully to human biology. The differences are enormous. There are species differences at every level of biological action, from your DNA to your cells and organs and how your whole body physiology functions, and even environmental influences that alter your cellular responses. These are very different between humans and animals. Some of these differences are confounded by other factors such as experimental disease induction. The way we induce diseases in animals using artificial methods is very different from how humans get diseases naturally. And even things like age differences, sex differences, and strain differences in these animals can affect how we interpret these data. And even animal husbandry practices and animal handling and how we treat them can all confound the way we apply these data from animal models to humans.
So we need a human-relevant system that will allow us to better predict how humans will respond to medications, to foods, and to chemicals. Right now, when we're developing drugs, we have two tools at our disposal: animal testing or cells in dishes. Because animals aren't humans, they're often failing to predict how humans will respond to drugs. And because the cells in the dishes don't work like they do in the human body, they're not very predictive either.
Monozygotic twins started from the same zygote. Okay, so they were genetically identical at the time of conception. You will never have an animal that is more genetically identical to a human than monozygotic twins. And yet we know that monozygotic twins do not always suffer from the same diseases. For example, one twin may come down with schizophrenia, the other one will not. One twin may have a cardiac disease while the other one does not. And even when they do have the same disease, for example, maybe they both have breast cancer, they may receive different treatments because of incredibly small genetic differences that occurred after the zygote split. So that should really inform us about how much knowledge we're going to be able to obtain using an entirely different species. I mean, if monozygotic twins, identical twins, cannot tell us what the other twin is going to do in terms of her reaction to drugs and disease, it's crazy to think you're going to get that information from studying a completely different species.
Rats and mice, because they're smaller, have a very high rate of metabolism. And as we know, drugs are profoundly affected by the metabolic environment that they're put in. And so that can impact the effect of the drug. And in some cases, we do see that the animals are able to clear the drug quickly, so there are minimal toxic effects. Whereas when we move to humans, those drugs stick around longer, they affect a greater range of cells, so their effects may be amplified.
Look close and you can see it beat. Heart tissue grown from stem cells used to test future drugs. The miniature heart-like structure suspended in a jar, pulsing. And most drugs never make it past testing. The main reason they are toxic to the human heart, something difficult to determine when testing on animals and often not discovered until well into the human trial phase.
Humans are as different from animals like mice and dogs and cats and kangaroos as they are from each other. The more effort we spend as a society learning about the animals, that effort could be applied to learning more about humans. And that's, of course, our goal, is to learn more about how we work from every scale, from the nanoscale up to the full organism, and how we interact with each other.
I think one of the issues that arises with newer and better technologies is we can look in more and more detail at any given organism. So the question becomes, how much can we study in animal models and how much is it applicable? And I think it's fair to say there is always the danger that you learn more and more about the mouse or the rabbit or whatever organism, but it may not apply to humans.
For example, with osteoporosis, a study was done to find out how effective two osteoporosis drugs were. It was found out that by trying to replicate that, that the mouse skeleton doesn't age the way the human skeleton does. So the human skeleton is just not comparable to a mouse skeleton.
I think some of the ones that are brought up frequently are polio, all the primate studies that were done that led to the production of vaccines for virtually the complete control of polio. I think that's a story that's been overzealously presented. There's a good deal of evidence that actually the use of primates slowed down discoveries of some of the most important aspects of viral activity. Studies on diabetes and the production of insulin requiring the use of dogs, I think gets a lot of credit. And I wouldn't say there wasn't some value in that work, but I think it's been overzealously described, and alternative for those same discoveries were available.
I'm Aisha Akhtar. I'm a neurologist and a public health specialist. I'm Deputy Director of the Army's Traumatic Brain Injury Program, and I worked for almost a decade at the Food and Drug Administration. And of course, I have to say that these opinions are my own. Researchers have been doing animal testing for decades, right? You can say a century or more. And what's amazing is that up until recently, no one really stopped to ask if animal testing is the best way to understand human biology and human diseases. And it's only been recently that some researchers and scientists have started to really critically examine animal testing and to see if animal testing predicts what we're going to find in humans. And again, these studies are being churned out, and again, these studies are showing that animal testing is overall very ineffective in helping us understand what we're going to find in human biology. There are three main causes for why animal testing can be ineffective. And the first, and the research has shown, that just about anything a researcher can do to an animal in the laboratory can cause changes in their physiology that we cannot predict and that we cannot control. So everything, anything a person does to an animal in the laboratory can change the testing outcomes. So we're talking about the type of flooring that an animal is tested on, the type of cages that they're in, the type of lighting in the room, the humidity level in the room, how often the animals are handled, how they are handled, what they're fed, what species they are, and what strain they are, even what distributor these animals were bought from. All of these factors can cause changes in an animal's biology that can cause misleading results, ultimately, in the tests that are being performed. So for example, studies have shown that putting non-human primates in one type of cage versus another causes differences in their neurochemistry and in their neurobiology. The second way that animal testing can be ineffective is that in the laboratory, researchers are trying to artificially induce human diseases in the animals. So for example, stroke occurs usually when there is a blood clot to a certain part of the brain. And it's a relatively simple disease in comparison to so many other diseases. But researchers have been trying to recreate stroke in animals for decades. And what they tend to do is that they tend to artificially induce a clot into a blood vessel into the brain, or they clamp down, they get a clamp that sort of closes a blood vessel to create this stroke artificially again. And the reason why is that most animals in the laboratory don't naturally have stroke. So in humans, for example, stroke is usually preceded by atherosclerosis, which is a narrowing of the blood vessels because of plaque buildup. Well, that's not happening in animals when we're clamping down on their blood vessels or we artificially insert a blood clot into their vessel. In humans, a risk factor for stroke is diabetes. Animals in laboratories don't usually have diabetes when being tested on for stroke drugs. In humans, stroke is often coincides with high blood pressure. We're not seeing that in animals in the laboratory either. We cannot recreate these human diseases which are occurring naturally, artificially in animals in the laboratory. And the third way that animal testing can be ineffective is simply there are just so many differences in our genetics, not just in our genetic makeup, but in how our genes are expressed, as well as our physiology, between species.
We've recognized substantially in recent years is the lack of reproducibility of the animal model. And there's actually been some great publications recently that if you run an animal test, you run it again, it may not even predict the first test. And so the reproducibility of some of these modern approaches are much, much better. The difficulty is that in validating the new approaches, we're always having to go back to those animal data as the gold standard and validate against those. But I've just told you that they're not reproducible. So that's a real problem that we have in validation. And what a lot of people are talking about right now is let's get away from comparing the new method to the old method, which we know has limitations, and instead compare the new method to what we know about human biology. Does it represent a pathway in the human biology that will lead to an adverse outcome? And if we can build confidence that the tools represent a step or several steps along that pathway, then we should be able to use that new approach and just move away from the animal.
Perhaps the main reason why people think that animal testing is so effective is because that's what we hear in the news. You know, if you go look through the New York Times, the Washington Post, or other major news outlets, we see again and again reports of breakthrough drug. You know, this new drug is going to cure this form of cancer, or a great new drug for spinal cord injury or traumatic brain injury. And what people don't realize is that these breakthroughs, so-called breakthroughs, were the results in animal testing, usually. And then ultimately, they end up failing when going into clinical trials and when tried in humans.
The manufacturer has actually quite a lot of leeway because they can switch species, rodent species. I mean, you know, you can use a mouse, you can use a rat, you can use a guinea pig, you can use a hamster. So they will switch and change until the result in the rodent and the non-rodent species correlate. They cannot proceed to clinical trials until they get that step right.
If you need, say, 2 animal species before you submit your drug proposal to the Food and Drug Administration, if you don't get the results you want in a mouse, you might try to get it in a rat. And if you don't get it in a rat, you may try to get it in a species of fish or some other lower animals. And if you don't get it in a dog, you may try to get it in a cat or a monkey. If the results you get depend on what animal you use, and you can move from animal to animal until you get what you want, I don't know how you could have better evidence that animal research is unreliable, completely unreliable. And that shows up when you start testing in people and you see that 95% of what you thought happened in animals, whether you pre-selected them or whether you changed along the way, doesn't apply to people. Doesn't really matter. You can make the animals tell you what you want, but it's not going to relate to what happens in people.
Developing a drug is a lengthy and costly process. On average, it takes about 12 years to develop a new drug. These 12 years do cost a company about $1.4 billion. $1.4 billion to get one drug to the market.
Kevin Costa is one of the scientists behind Novoheart.
The drug discovery process is a lengthy and expensive process. It typically takes about a decade and a couple of $1 billion US to take a drug from its initial concept to clinical trials.
Costs are crazily shocking. Well, I estimate, I think I'm out of pocket probably somewhere around $10,000 to $12,000 for medications and supplementary treatments. And that's even with the federal government supplementary health care. The one I'm about to go on is somewhere between $40,000 and $60,000. But I have run into other friends that are going through this that have said they've actually had to sell their houses to pay for the medications.
We're not investing as much, and I think the innovation that we see, some of the really cutting-edge non-animal or human-based approaches, are not being pioneered here.
There will be missed opportunities, and more importantly, people will not be able to be part of what we see with the rest of the world as advancements with regards to medicines, treatments, and a number of different research innovation items.
The move away from animal testing does not imply that we're suddenly going to be shifting to testing on humans. That's not what's being proposed at all. The non-animal approaches, the human-based approaches, are often based on, say, three-dimensional tissue, such as skin or eye, which are from surgical waste. There are a number of ways that ethically sourced human tissues are available, and that's what we're talking about, is using and creating models that are relevant to a particular situation. So we can take bioengineered organs on a chip as an example.
The lung on a chip is a microdevice that has the potential to replace animals for the testing of drugs and toxins. The device, which is lined by human cells, mimics the mechanical and chemical functions of a living, breathing lung.
Every funding dollar, every taxpayer dollar, every charitable dollar that goes into medical research, a decision could, and I would argue should, be taken: is this research going to be relevant to the human species?
We're missing out on opportunities, we're squandering resources because we're based upon an old model that is very defensive about protecting its turf. And change can be scary, not only just to anyone who's supplying in this chain of the money exchanges, to the business that gets done, to the research of papers and institutions and private industries, but they all receive this opportunity at the forefront, is that Canada can be actually part of something new. It's, the fact is that we're behind and can catch up and play an important role.
This is real modern 21st century science, and to be able to replicate, say, lung research, respiratory toxicology, using these tools rather than sticking rats in an inhalation chamber for hours a day and having results that are actually predictive of the human situation, it's a no-brainer.
When researchers are looking at new chemical compounds that may eventually become drugs, for example, they're looking at, on average, 5,000 to 10,000 drugs. So let's say you start with 5,000 to 10,000 drugs, and out of those, only about 5 end up going into human trials. So a lot of drugs were abandoned in that process, or a lot of potential drugs were abandoned in that process. We may have been abandoning potentially very useful drugs that may have been very effective, or maybe even been cures in humans.
We actually don't know whether those drugs do harm in humans or do nothing or actually are effective. So really, it reveals the fact that independent of how much animal testing there is, we still need to do human testing. So it doesn't really directly help, and actually could hurt by giving us the wrong information.
Firstly, we may have lost drugs that may have worked in people that didn't work in animals. The second point is that testing on animals gives researchers a false sense of security, if you will, because something that appears to work in an animal may then not work in people, may cause harm. And in fact, we have lists and lists of drugs that were apparently safe and effective in animals and then did a lot of harm in people. Now, according to the FDA, for every 10 drugs that pass animal tests, 9 out of 10 will fail during human clinical trials.
More than 90 drugs, vaccines that have been tested in animals for HIV have failed. More than 100 drugs that have been tested for stroke in animals, which worked in animals, failed when tried in humans. More than 150 drugs that were tested in animals to treat inflammatory diseases and worked in animals failed when tried in humans. More than 20 drugs that were tested in animals for ALS failed when tried in humans. More than 27 drugs that were found effective in animals for treating traumatic brain injury failed when tried in humans. Repeatedly, drugs that have been found safe and effective in animals have failed when tried in humans.
When there is correlation between what is found in a particular animal and what is found in people, it's random and uncommon. There are many examples of delays or prevention of finding treatments and cures for diseases because animals were used. It has delayed or prevented us from finding therapeutic approaches that work for many serious chronic diseases like Parkinson's disease or Alzheimer's disease, neurological disorders, and a variety of other conditions. And that's why of all of the hundreds of human diseases we have, effective treatments or cures for so few of them.
Experimenting on animals is slowing down progress because I think there's a lot of time wasted, a lot of resources that are burned on developing a new mouse model, a new rat model, a new genetic model in a rodent species or in some other non-human animal species to try and understand disease when at the end of the day, it's not as relevant as a model that uses human biology. And so a lot of time and energy and money and resources are going into animal experimentation, and that is really diverting the focus from human relevant science, which is where it should be. So I think it is slowing down a lot of progress in terms of understanding disease mechanisms, understanding chemical toxicity, understanding things like human genetic susceptibility to environmental exposures and into diseases.
The reminder that we have, particularly in the drug area, is it begins with in vitro testing in cells and tissues, not terribly sophisticated currently. It moves on to animal models, rats, mice, dogs, pigs, monkeys, then moves on to humans. Why does it move on to humans? If we really trust the animal models, why do we need that human clinical step? The truth is we don't entirely trust the animal models.
As a matter of fact, about 20 years ago, the Food and Drug Administration had admitted that about 9 out of 10 drugs that are found to be working in animals end up failing when tried in humans. And more recent analysis suggests that that failure rate is even higher. It's even closer to 96%. So not only are we not improving, we're getting worse. And this is because we are continually investing in animal testing when we need to really start looking towards the future and start thinking about how best to study human biology. And that is through human-based testing methods.
So imagine someday in the future, you walk into a doctor's office and the doctor scrapes a few cells off your skin, and those are sent to a research lab where they're grown and expanded and reprogrammed into any cell type in your body, whether it's brain, kidney, liver, heart, lung, or pancreas. And these cells carry your disease. They mimic what happens in your body. And these cells are then turned into miniature organs through tissue engineering and bioprinting technologies, and those miniature tissues are put on platforms like organs on a chip, where hundreds of drugs can be tested to select the one that's most appropriate for you, the one that would respond to your cells, your biology, your disease, not mouse biology.
What we want to do is to be able to predict what a medicine is going to do to you before you take it. Right now, we don't really know what medications are going to do to you. A, it might not work. B, you might have a very bad side effect to the medication. So what personalized medicine does is it tries to match your genes to very specific drugs. And so if we know what your genes are and we know what those genes do in terms of this medication, we can make sure that you get a medication that is both safe and effective. This is the holy grail of medical practice. It always has been. And this is not futuristic. We are doing this. There are over 150 drugs available right now that before you take one of those drugs, you will have your genome profiled. We will figure out if you have the right genes for this or if you have genes that are going to render the medication ineffective.
Precision medicine is the concept of developing treatments that are specific to one person. So by definition, they couldn't really be tested on other people or animals or anyone. If we have a detailed internal view of the body from an MRI, we can do surgery planning. So we can plan specifically how to fix something or 3D print parts that are needed inside that are important to have a successful surgery. For example, in jaw surgery, it's very important to have symmetry, that you can close your mouth and speak and swallow and everything like that. If it's a medical treatment, a drug, or some kind of other treatment that might involve your own cells, a sample could be taken, like a blood sample, and that can be replicated and reverted to stem cells to develop specific types of cells that you might need, and also to test different approaches to see if they'll be effective before they're even tried on you as well. So try it on the sample.
My name is Dr. Charu Chandrasekhar, and I'm the founder and executive director of the Canadian Centre for Alternatives to Animal Methods and its subsidiary, Canadian Centre for the Validation of Alternative Methods, located at the University of Windsor. Our centre is the first of its kind in Canada. The overarching vision of the Canadian Centre for Alternatives to Animal Methods is to promote the replacement of animals in Canadian biomedical research, education and regulatory testing through 21st century science, innovation and ethics. At the undergraduate level, we're creating courses that we hope to develop into minor and major degree programs. At the graduate level, we're creating a one-year master's program in animal replacement science to teach students everything from human biology, human-centered experimental design, and animal welfare and bioethics. And we're also training doctoral students in our facility.
The reason why people seek themselves and why they want to work here at CELINC is because we're truly changing the world of medicine. And being able to be part of that story is a tremendous impact, both from a personality perspective, but also from the perspective that you're doing something that can truly impact the world and the generations to come. The company focuses heavily on developing novel technologies that enables researchers and scientists to print human tissues and organs.
CELINC is the first bio-ink company in the world and also leading provider of bioprinters. Our business platform and our business model is to provide scientists and researchers with tools so they can print human tissues and organs in their labs.
I'm Erin Hill. I'm the president and co-founder of the Institute for In Vitro Sciences. IIVS is a nonprofit organization, and we offer in vitro, which are non-animal testing services, to a variety of industries. And we also have educational and outreach programs for government and different agencies around the world to accept these methods.
We call these organ chips. Each chip is engineered to create the right environment for cells to feel like they're within the human body.
The chips provide the mechanical forces that are critical for the cells. When you breathe in and out, you can feel the forces that the cells experience as your lungs expand and relax. There is also the flow of blood that creates a dynamic environment and brings in nutrients and takes away waste products. All of this taken together allows us to recreate the microenvironment that the cells need so that they can behave just like they do in our human body.
My group develops protein engineering to develop potential therapies for various diseases. We're very interested in exploiting modern methods of genomics to address diseases including cancer. And our fundamental belief is that the more we can understand about the signaling networks that drive normal cell growth, the more we can then find why those are aberrant in cancer. And then crucially, we develop new molecules or proteins that can go in and fix the problems in the signaling. So we take a very molecular-based approach to disease.
I'm a biomedical researcher. My work focuses on trying to understand how stress during pregnancy affects the development of the fetus. In the process of understanding that, we've come across some really interesting challenges in understanding human biology. Specifically, my work focuses on the placenta, and trying to model the placenta has become a real challenge because there are no good animal models of the human placenta. And so one of the directions my lab has taken is to really try to be pioneers in developing an in vitro or out-of-the-body model of the human placenta. The aim is to be able to bring together different types of human cells that build organs in combination with the environments that they live in, and be able to model even the air, the oxygen concentration that they experience, the effect of the blood rushing by the tissues. So all these parameters are not usually reflected in the existing types of testing models that we do. And as we build these more complex models, there needs to be a focus on considering all of these different factors, looking at their biological response and taking those responses into consideration as we test different drugs.
The Parametric Human Project is a nonprofit I started in 2014 with a number of professors at different universities with the goal of developing the most advanced digital human model possible. There are several disorders that involve the musculoskeletal system. The innervation, the nerves, and us being able to control how we use our bodies is just one system in the dozen or so systems in the human body. This really is a slice of the systems biology effort to understand how all the human systems relate to each other and affect each other. That broader approach of systems biology is what we need, and we need to complete our understanding of all of the systems, including the musculoskeletal system, to have a complete view of the body, of how even drugs are distributed throughout the body, for example, through the vasculature in the muscles. That's the primary way that a drug will go to the brain or anywhere.
We have built in this process is called the virtual physiological human. It is a new technology that makes possible to capture, finally, the holistic aspect of the human physiology and pathology. But before this has an impact in healthcare, there is one little magic more that we need to bring in. If I now feed to my virtual physiological human simulation the data relative to a particular individual, now that simulation will make prediction about the health status of that individual. This is not personalized medicine. This is individualized medicine. We can finally say something about not you because you are about the same age and the same sex and the same disease of another 1,000 of people, but because you are you, with your condition, with your history.
The area that we've been most excited about is using computer simulation to reproduce what's going on when a drug causes bad things to happen in an organ. And we've been particularly focused on liver, since that's certainly one of the most problematic adverse events that can occur, sudden liver injury. We take the drug and measure how the liver is exposed to it, and then reduce the processes whereby the drug can hurt the liver into differential equations, and then can combine all that into a predictive computer-based model. And what you put into the model are the effects of the drug, and sometimes the breakdown products and metabolites, on certain pathways and processes that can be measured in the lab. And none of this involves animals, and that's one of the most exciting things about it, is that this technology is really delivering on the promise of predicting drug safety without having to test the drug in a variety of animals, usually at least rat and dog, but often rat, dog, and monkey, really to satisfy regulatory requirements. It certainly holds the potential to being much faster, and I think also much more economical. So I think it really is paving the way to more efficient development of new drugs.
You know, we shouldn't really be looking at alternatives only just replacing existing animal models, but we should see it as an opportunity to tackle problems which we haven't been able to tackle effectively in the past. Look at a whole area of neurodegenerative diseases, for example. We simply don't have effective animal models to deal with that. So it's not about replacing in that sense. It's about providing something new, providing a new opportunity to do human relevant research and ultimately develop those treatments and understand that human biology that we can't really do with traditional approaches.
I think if I were a parent, I'd be very concerned with just the state of science. Everything in every other part of our lives, we embrace the new technologies so readily, so hungrily. Yet, why is medical research still using models developed, in the 1980s or in some cases earlier?
In government, things can move slowly, and we need citizens when they're passionate about an issue, whether it be the humane treatment of animals or whether it be advancing our health system so we have more precision in our healthcare system and more individualized care. We need experts and citizens alike to be coming together and pushing this onto the government's agenda.
Unless we, with focus, drive to being a leader in this field where we have in our midst companies, resources that will attract investment, we're going to be losing people, we're going to be losing chances to generate revenue for our federal budgets. For companies that come to Canada, they're going to start looking elsewhere.
I know of many, many grants that are based on excellent science that are routinely rejected because they don't include animal tests as part of their proposals.
It's crazy to reject applications that don't test on animals if at the same time we're not investing in research to move away from testing on animals.
People who are forward-looking in using human relevant approaches often hit a wall when they look for public funding because those who are judging them are locked into the paradigm of animal testing. And it seems curious, but it's true that even if you're addressing a potential advance in humans, many times your protocol will be denied, will be given a very bad score and won't be funded because you're not doing it in an animal model that other people can use to study the disease.
When contacted, both the government and charities were unaware they were denying grants for alternative methods.
Canada is lagging behind. Canada has not invested in this field scientifically, financially, or legislatively as many other countries have. From the Americas to the Far East, countries across the globe have already established national, federally funded centers dedicated to alternatives and brought in legislation and national strategic roadmaps to shift away from vertebrate animal testing. But we're nowhere near there. We just started our first center a year ago. Canada needs to catch up.
In 2017, the Canadian Centre for Alternatives to Animal Methods was the first of its kind to open in Canada. To date, the centre still receives no government funding.
There's no doubt that Canada needs to pay more attention to this and actually have more political resolve. That's what it's going to take. The reality is that government can show leadership because we have to deal with industry interests, supply management interests, scientific and institutional being research areas that have interest, all that are receiving grants, monies, financial incentives, and empowerment models that are based upon decades of an establishment. And to shake that foundation in a positive way will require government leadership.
We live in a democracy and it's representative democracy, and so it's contingent and dependent upon citizens taking the lead and pushing issues onto the agenda by meeting their members of parliament and meeting members of the government.
I've been petitioned by thousands of Canadians to make sure that in Canada we move forward and ban the testing of drugs on animals. It's not necessary. Anywhere you look, there's a lot of weight against progress that is wrapped up in the status quo. And the more that people push for change, the better.
Inform themselves of the issue, inform themselves of the fine print, and contact policymakers, contact their elected officials, contact the charities they support, and ask, if I make a donation, how is this money going to be spent? Are you really investing in these new technologies? Are you supporting a future, or are you simply funding the same old using animals? And the same is true of policymakers who make decisions on how the federal funding agencies spend their budgets, our tax dollars. It's really up to individuals. We have the power, it's our money that's being spent, but we need to take the time to understand the issue, understand the complexity. It's not simple, it's not black and white, but the truth is we can do much better than we are, and we need to break out of that, the animal model paradigm.
The best thing that the public can do is demand more from their politicians. We are all, each of us, are paying for animal testing through our tax dollars and sometimes through our donations. Studies are showing animal testing is just so largely ineffective. If we want to protect our children, we want to protect our loved ones, we need to demand that we start using better testing methods, and that's not animal testing.
Two branches of government, Health Canada, Canadian Institutes of Health Research, along with the Canadian Council on Animal Care, all refused to be interviewed for this film. The Canadian Cancer Foundation, Parkinson's Canada, the ALS, Diabetes, and Alzheimer's Societies also ignored our request for interviews.
To date, charities do not offer a link to donate to alternatives. Canadian universities continue to support and encourage an outdated system to students, and our government provides minimal funding towards human biological innovations.